Welcome to the Deep Dive. We're here to take really fascinating, sometimes complex, stacks of research, break them down, and give you that pure, distilled knowledge. Today, we are unlocking one of the body's most intricate and foundational systems, the endocannabinoid system. You might know it as the ECS. Now, if you've heard of the ECS, you probably link it straight to, well, the cannabis sativa plant. But honestly, that's like seeing one really famous tree and missing the entire forest. This system, it's your body's own essential ancient balancing mechanism. It's not just tweaking your mood or helping you sleep. It is constantly working behind the scenes, adjusting everything, really. Pain, inflammation, metabolism, even how your brain handles stress.
It really is foundational. And what's fascinating, truly fascinating, is the evolutionary perspective. The appearance of these endocannabinoids are internal ones in vertebrates. That predates the cannabis sativa plant by something like 575 million years.
Well, 575 million. So this system was running on internal signals way before.
Exactly. Long before humans ever encountered the plant. So this isn't some minor pathway. It's absolutely fundamental to physiological homeostasis, keeping everything in balance. It's core to vertebrate health.
Okay, so for this deep dive, our sources are pretty broad. We're looking at cutting-edge stuff like acute brain imaging, including that big Canteen study on young cannabis users. But we're also looking at global regulatory views, like from the CDC, on therapies, research gaps, and really critical things like drug interactions and workplace safety.
Yeah, and our mission here for you listening is to be really comprehensive. We need to clearly lay out the components of this huge internal signaling system. Then we need to analyze the hard neurobiology, what actually happens acutely when external cannabinoids like THC and CBD hit the brain. And finally, and this is crucial, unpack why, despite decades of scientific promise, getting these compounds into regulated therapeutic use is still, well, it's startlingly complex. We have to understand that tension, that push and pull between the pure pharma approach and the reality of these complex plant extracts, sometimes called informally dirty drugs. Let's dive in.
Okay, let's start with the basics, the chemical foundation. There are three core components to the ECS. You can kind of think of it like a signaling system that works backwards. It's this finely tuned feedback loop, right? Designed to dampen down excessive activity. So what are those three parts?
Well, first, you have the infrastructure, the cannabinoid receptors. The two big players everyone talks about are CB1 and CB2. Now, CB1 is everywhere in the brain, and crucially, it's one of the most abundant receptor types in the entire mammalian central nervous system, especially in areas that govern things like memory, motor control, mood.
So its heavy presence in the brain is why.
Exactly. That's precisely why external cannabinoids, like THC, have such powerful psychoactive effects. They're hitting this incredibly widespread system.
Got it. And the other one, CB2, is that also in the brain?
CB2 is primarily found outside the central nervous system. Think peripheral organs, and are really notably on immune cells. So it plays a big role in things like inflammation and immune responses. But it's not strictly peripheral. There's evidence that CB2 expression can actually go up in the brain under certain conditions, typically bad ones like neuroinflammation or injury.
Yeah.
So you could say maybe CB1 handles the sort of moment-to-moment brain signaling, while CB2 comes more into play when the body's under stress or attack.
Right, so those are the docking stations, the receptors. What's the second component, the things that actually dock there? The body's own signals.
Correct. Those are the endogenous ligands, the endocannabinoids our body makes naturally. The two most studied are anandamide, or AEA, sometimes called the bliss molecule. Maybe you've heard that.
Yeah.
And the other major one is 2-arecanyl glycerol, or 2-AG. Now, anandamide, AEA, is a partial agonist at CB1. It gives a sort of gentle nudge. 2-AG, on the other hand, is generally seen as a full agonist, especially for CB2. And interestingly, it's present in the brain at concentrations maybe 50 to 100 times higher than anandamide in certain regions.
Wow, much higher. So what's really unique about these molecules compared to, say, classic neurotransmitters like serotonin or dopamine?
The key difference is really the timing and direction of the signal. Classical neurotransmitters, they're stored up in little packets, vesicles, right? And released from the presynaptic neuron to signal forward across the gap, the synapse. Endocannabinoids are different. They're synthesized on demand, typically in the postsynaptic neuron, the receiving one, and they travel backward across the synapse.
Backward.
Yeah, retrograde signaling. It's like they're acting as a chemical dimmer switch. They essentially tell the presynaptic neuron, hey, slow down, you're firing too much. It makes them perfect regulators for maintaining stability, keeping things quiet.
Okay, that backward signaling is cool. And the third component, you mentioned it's critical for keeping things localized and fast. The off switch.
Exactly, the enzymes. They degrade these endocannabinoids really quickly once the message is delivered. This speed is absolutely essential. The two key enzymes are FAH, fatty acid amide hydrolase, which rapidly breaks down an anandamide, and then there's Magiel monoacylglycerol lipase, which is the main one for degrading 2-AG. Because these enzymes are so active, the endocannabinoid signal is super localized and very short-lived. If they weren't broken down quickly, the system would just be constantly on, leading to, well, functional chaos.
Right, you need that precise control. Now, when you look at the list, the sheer list of physiological processes the ECS controls, it really does feel like a master regulator. Can we just touch on the scope of what this system governs?
The scope is, honestly, breathtaking because it's involved in balancing so many different functions. Things like metabolic processes influencing fat storage, nutrient uptake, it modulates motor function, it's central to pain perception, nerve protection, controls appetite. The munchies phenomenon, that's a classic example of external compounds hijacking this natural system. And importantly, it's a key player in developmental biology and synaptic plasticity, the brain's ability to learn and change.
And that deep regulatory role, that brings us to something really interesting the sources highlight, the ECS and stress. How does it handle stress?
Yeah, connecting this to the bigger picture, the ECS is fundamental to how we start, how we process, and critically, how we stop a stress response. Research shows that when we experience acute stress, our stress hormones, particularly glucocorticoids, they rapidly increase endocannabinoid levels within the limbic system. That's the brain region key for emotion and fear memory.
So it's like an internal brake, a
Right.
calming mechanism kicking in.
Precisely. The ECS acts as that internal brake. ECS signaling, especially up in the prefrontal cortex, your brain's CEO, basically it contributes significantly to stopping the stress response in a timely way. If the system is impaired, maybe the FAAH enzyme is too active and breaks down anandamide too quickly, that stress response might just linger. And that could potentially lead to things like anxiety and mood disorders, which actually sets us up perfectly for the next section. What happens when we overwhelm this finely tuned braking system from the outside?
Okay, so if the ECS is our natural balancer, what happens when we, well, dump external cannabinoids into the system? Compounds that are often much more potent, stick around longer than our own AEA or 2-AG. We've got a really fascinating study to help us here, the Canteen study. They use fMRI, functional MRI, to look at this in young adults and adolescents.
Yes, this was a really important study because it looked at a very relevant group, semi-regular users, people using cannabis, say, between half a day and 3 days a week. The average ages were around 27, almost 28 for the adults, and about 17 for the adolescents. So this isn't looking at chronic heavy use, but more typical patterns in young people. They gave the participants the drug using vaporization, either THC alone, THC plus CBD in a 1-to-1 ratio, or a placebo.
And the timing of the scans was deliberate, right, to capture the peak effects.
Absolutely. They did the resting state fMRI scans about 50 minutes after administration started. That window is pretty well known to capture the peak subjective effects from vaporized cannabis. They really wanted to see the brain's immediate maximal response.
The way they looked at the brain was quite specific, too. They focused on 4 key networks, right? These big interconnected cortical networks involved in high-level thinking. Why those 4?
Yeah, they were looking for changes in how different brain regions communicate. The 4 networks were the default mode network, or DMN, that's active when you're introspecting, mind wandering. The executive control network, ECN, crucial for decision-making, working memory. The hippocampal network, vital for memory, obviously. And the salience network, or SN, and that's the one we really need to focus on here. They use specific landmarks, seed regions to define these networks, like the posterior cingulate cortex for the DMN and the anterior insula for the salience network.
Okay, let's pause there. The salience network, you said that's the key one. The study found the strongest effects were right there in the SN. Why is that network so important? What does it do?
Right. The salience network, with its hub in the anterior insula, basically acts like the brain's internal triage officer or maybe a spotlight director. Its job is to detect and integrate important or salient stimuli. That could be internal things like hunger or pain, or external things like a loud noise or seeing a face. Then it decides which information needs our attention right now, and it helps shift resources away from other networks like the DMN or ECN to focus on that important thing.
Ah, okay.
The fact that the strongest acute effects of cannabis were seen here in the SN tells us something really important. It suggests cannabis is primarily messing with our attention and prioritization mechanism. It's not just about memory or general executive function in this acute phase. It's about what we pay attention to.
So what actually happened to the salience network's connections when people took cannabis in this study? Did it get stronger,
Yeah.
weaker?
Weaker. Cannabis administration, and this was true for both THC alone and the THC plus CBD combo, significantly reduced connectivity between that salience network hub, the anterior insula, and several other important brain regions compared to the placebo group. These regions included areas involved in processing sensory information, like the temporooccipital cortex and the sensoromotor cortex, as well as the planum temporale. So it looks like a dampening or disruption of the system that normally directs our attention outwards.
Which fits perfectly with the subjective feeling of being high,
Mm.
right? Feeling less tuned into external stuff, maybe time feels slower, more inward focused.
Exactly. It aligns very well with those reported subjective experiences. But here's where it gets really, really interesting. The interaction between THC and CBD, did adding CBD just sort of cancel out the THC effect?
Yeah, that's often the assumption, isn't it?
Yeah.
CBD buffers the high.
Well, not here, not in this specific network measurement. This is the crucial finding that kind of complicates things. The THC plus CBD combination actually reduced connectivity even more significantly than THC alone in several key areas, specifically the sensoromotor cortex, the insula itself, and the planum temporale.
Wait, so adding CBD amplified the reduction in connectivity in those attention and sensory pathways. It didn't lessen it.
Correct. In these specific pathways, CBD didn't just mitigate the effect, it seemed to enhance the THC-induced reduction in connectivity.
Wow. That's a huge finding. For researchers, for consumers,
Yeah.
it really hammers home that CBD isn't just some inert buffer you add for marketing. It's pharmacologically active, changing the whole picture. But why? Why would it amplify that effect?
Yeah, the exact mechanism isn't fully pinned down yet. It really speaks to the complexity we keep talking about. CBD hits dozens of targets in the body. It's not just an anti-THC molecule. It might be changing how THC binds to the CB1 receptor, maybe altering its shape slightly. Or it could be affecting how THC is absorbed or broken down, maybe letting more of it reach those specific brain areas. Or perhaps it's acting on totally different receptors, like serotonin receptors, that then indirectly influence the salience network. The main takeaway for you listening is that when you mix these compounds, you're creating a whole new neurobiological equation, and the results can be really counterintuitive sometimes.
So if we zoom out a bit, what does this overall acute reduction in connectivity, especially in the salience network, suggest about the state of intoxication itself?
Well, some other studies using whole brain approaches have also suggested this kind of widespread hypoconnectivity, less connection across various networks, particularly subcortical, limbic, and attentional ones, during cannabis intoxication. And what's kind of interesting, perhaps analogous, is that this pattern of acute change where brain networks seem less integrated, less talking to each other in their usual organized way, it bears some resemblance, structurally at least, to the effects seen with classic psychedelics like psilocybin.
Really? Like LSD or magic mushrooms?
Yeah, though likely through very different downstream chemical pathways, of course. But both classes of compounds seem to acutely disrupt the brain's habitual, organized communication patterns. And that disruption might be fundamental to the altered state of consciousness users report. All right, let's shift gears now. Moving from those acute brain effects towards the clinical potential, the therapeutic landscape. And this is where, the scientific promise feels immense, but the gap between exciting lab findings and actual human medicines, well, it becomes incredibly clear, stark even.
Yeah, let's start with chronic pain. It's still one of the biggest public health challenges out there, and it's always been a major target for trying to modulate the ECS, right?
Absolutely. And the preclinical support, the studies in animals, it's overwhelming. There's substantial evidence from established rodent models, things like the formalin test for acute pain, or the Complete Friends adjuvant model for inflammatory pain, showing that cannabinoids and other ECS modulators have real potential for developing new painkillers, new analgesics. Researchers are really focused on trying to boost the body's own system, enhance those natural endocannabinoids, while specifically avoiding the psychoactivity, the high, that comes from directly hitting the CB1 receptor too hard.
Okay, so how do they plan to do that? Boost the system without the high?
Well, one major strategy is targeting those off-switch enzymes we talked about rather than the main receptor itself. So they're developing drugs that inhibit FAAH, the enzyme that breaks down anandamide, or inhibit MGL, the one that breaks down 2-AG.
Ah, so slow down the breakdown.
Exactly. The idea is to let your body's own calming endocannabinoids hang around in the synapse a bit longer, do their job better, but hopefully only locally and gently, not causing a big global effect. Other approaches include developing agonists, activators specifically for the CB2 receptor, which is mostly outside the brain, or developing things called CB1 positive allosteric modulators, or PAMs. These don't flood the receptor like THC, they just sort of subtly fine-tune its activity.
Okay, those sound like really clever, targeted approaches. Non-psychoactive pain relief is like the holy grail. But you mentioned a gap. Despite all these promising pathways, you said over 150 preclinical studies, the sources point to this enormous, almost unbelievable gap when it comes to human trials.
This is honestly one of the biggest shockers in the research stack we reviewed. And it really highlights why the regulatory complexity is such a huge issue. We have, yes, over 150 robust preclinical studies looking at CB2 agonists, endocannabinoid reuptake inhibitors, AirGL inhibitors in animal models of pain, showing promise. Yet, as of the data we looked at, there are 0 randomized controlled clinical trials, RCTs, for the first 3 of those drug classes in actual human pain patients. 0.
0. After 150 plus animal studies, that's staggering.
It is. For FAH inhibitors, there have only been 3 completed RCTs looking at their effectiveness in humans for pain. So a massive preclinical effort, years of work, millions invested, and almost none of it has translated into testing these potentially non-psychoactive options in the people who need them.
Why? What's the bottleneck? We have this huge need for non-opioid pain relief. We have promising science. Why is it stuck?
It really seems to stem from a combination of factors.
Yeah.
There's the lingering shadow of the legal classification of cannabis itself, which historically made research difficult and maybe deterred some investment. And then there's just the inherent risk and cost of drug development. Moving a drug from early safety trials, phase 1, through efficacy testing, phase 2, and then large-scale comparisons, phase 3. That takes billions of dollars and many, many years. A lot of drugs simply fail in phase 2 or 3, either because they don't work well enough or they have unexpected side effects.
Right.
So because this whole field started from a legally complicated place, it's been running uphill. It's dramatically slowed down the translation of that promising preclinical science into regulated medicines people can actually access. Meanwhile, of course, people are self-medicating with unregulated products.
Okay, let's turn to another major area, cancer. Both understanding the basic biology and also managing symptoms for patients.
Yeah, the preclinical insights here are also really compelling. There's evidence that activating cannabinoid receptors can influence core processes involved in cancer, like how cells multiply, whether they undergo programmed cell death apoptosis, and how tumors grow new blood vessels to feed themselves, angiogenesis. Specifically, there were studies mentioned on human glioblastoma cells. That's a really aggressive brain cancer. They showed that THC and CBD, especially the combination, could inhibit tumor growth and also suppress mitochondrial respiration, basically, choking off the energy supply the tumor needs to survive. Pretty significant findings in the lab.
And moving towards helping patients dealing with cancer now, particularly with cancer-related pain, which can be just awful and hard to treat. Is there clinical evidence there?
Yes, here we actually do have a medicine that has made it through the regulatory hurdles, at least in some parts of the world. That's nabiximols, which is sold as Sativex. It's a pharmaceutical-grade spray used under the tongue or inside the cheek, containing a precise 1.1 ratio of THC to CBD. A systematic review focused specifically on cancer-related pain found that nabiximol showed evidence of pain reduction in 5 out of the 6 clinical trials they analyzed.
And using that oral spray route rather than swallowing or inhaling, that's important for how the drug gets absorbed, isn't it?
It is key. It bypasses what's called first-pass metabolism in the liver. When you swallow something, a lot of it gets broken down by the liver before it even reaches your bloodstream. The oral mucosal route avoids much of that, which likely improves the delivery and means you lose less THC before it can get to work.
Makes sense. What about side effects in those clinical trials, even with a pharmaceutical-grade product?
Well, the most common side effects reported across the trials were generally things considered manageable. Nausea, some vomiting, dizziness, things you see with many medications. However, some patients did still experience psychological symptoms, things like paranoia, euphoria, anxiety, sometimes insomnia. These seem to be reported more often with inhaled formulations used in other contexts, suggesting that the rapid delivery and higher peak blood levels you get from inhaling might increase that risk. So even in a controlled medical setting, those psychological side effects are still something to watch for.
Okay. Now, for some of those real aha moments, particularly in the neuropsychiatric area, let's talk anxiety
Yeah.
and how our own body manages it.
This is fascinating. There's a really clear natural protective role for anandamide, our internal bliss molecule. Preclinical models, animal studies, consistently show that if you increase anandamide levels, which happens naturally after exercise, for example, or if you block that FAAH enzyme that breaks it down, you get clear anxiety-reducing effects, anxiolytic effects. And crucially, these effects depend on the CB1 receptor working properly. It's mediated through that system.
And this translates to people.
It seems to. There's a specific gene variant, the FAAH385A allele. People who carry this variant have slower breakdown of anandamide, meaning they naturally have higher levels of it just circulating around. And studies show these individuals tend to self-report lower levels of anxiety.
Wow. So if you're genetically wired to have more anandamide, you might be naturally more resilient to anxiety.
That's cool.
And you mentioned earlier that CBD seems to tap into this pathway somehow.
Exactly. There was a randomized clinical trial looking at cannabis use disorder, and they found that giving people CBD actually increased their anandamide levels.
Now, did they figure that out?
The exact mechanism isn't fully clear from that study alone. It could be related to CBD inhibiting FAAH a bit, or maybe changing how anandamide is transported across cell membranes. But the finding itself is really interesting. It suggests CBD isn't just working on its own targets, but it might be actively boosting our native anti-anxiety system, our endocannabinoid system. It reinforces that idea of it being a potential bioenhancer.
Okay, finally in this section, there's a really specific kind of mind-bending animal study finding about memory. It introduces this complex idea of state-dependent learning. This one took me a minute to wrap my head around.
Yeah, this is a very specific, very intriguing study. It used a standard fear conditioning model in rats' passive avoidance memory. Researchers injected a non-selective CB1 agonist, a drug called WIN 55212-2 that strongly activates CB1 receptors directly into a specific brain region called the central amygdala, the CEA.
Yeah, and the amygdala is key for fear learning, right?
Exactly. Crucial for emotional learning and consolidating fear memories. Now, they did this injection after the rats had already learned the fear task. And what happened was this post-training injection caused dose-dependent amnesia. The rats basically forgot the fear conditioning, shown because they weren't avoiding the shock zone anymore.
So the drug wiped the memory, seemingly. But that wasn't the end of the story. There was a twist.
Right, this is the key part. The amnesia wasn't permanent, and it wasn't because the drug had damaged the brain cells. It was state-dependent. They found they could completely reverse the amnesia simply by giving the rats another injection of the exact same drug, WIM 55212-2, right before the memory test.
Wait, giving the drug again restored the memory it had previously blocked.
Precisely. The memory wasn't actually destroyed. It seems it was stored in a very specific cannabinoid-influenced chemical state. The memory only became accessible again when the brain was put back into that same chemical state induced by the drug. It's like you needed a specific chemical key, the drug itself, to unlock the memory vault where that fear memory was stored.
That is... wow. That's a staggering finding. It implies these compounds don't just block or erase information. They can fundamentally change the context or the state required for retrieval, maybe through interactions with other systems, like adrenaline receptors.
Exactly. And it raises huge questions for human research, doesn't it? Especially for conditions like PTSD, where extinguishing fear memories is a therapeutic goal. If a cannabinoid-based treatment helps someone forget or extinguish a traumatic memory, is that extinction permanent and stable? Or could returning to a specific physiological state, maybe even just being stressed or intoxicated again, accidentally unlock or reinstate the original traumatic memory?
Yeah.
It shows just how deeply conditional these interactions with learning and memory can be. It's way beyond just feeling intoxicated.
Okay, let's shift now to probably the most talked about concept in the commercial cannabis world, but also one that's pharmacologically, well, let's say controversial, the entourage effect. Can we unpack this term? What did it originally mean versus how it's used today?
Right. The term entourage effect actually has a very specific scientific origin. It was coined in a preclinical study way back to describe how certain other molecules the body makes, which are themselves bioinactive, could sort of help out or potentiate boost the activity of a main bioactive endocannabinoid like anandamide. It was an internal finding about synergy within our own native system.
But that's not really how you hear it used in dispensaries or online, is it? Today, it's almost always used to argue that full spectrum or whole plant cannabis products are better than using just pure isolated compounds like synthetic THC or pure CBD isolate.
That's the modern market definition, yes. It argues that the complex mix of all the different cannabinoids, terpenes, flavonoids found in the plant work together synergistically to produce a better overall effect, maybe with fewer side effects, than any single compound alone.
So critics say it's vague, maybe just marketing hype, while advocates point to user experience.
Pretty much. Critics argue the term lacks precise scientific definition in this context and is mainly a marketing tool. Advocates, often citing anecdotal reports or patient experience, claim they feel a better overall therapeutic outcome or fewer adverse effects from these complex extracts. Pharmacologically, though, we should probably be using more established terms. We're really talking about synergistic interactions, where the combined effect is greater than the sum of the parts, or bioenhancement, where one compound helps another work better. Essentially, it's polypharmacy using multiple drugs or compounds at once.
Okay, let's focus on the most studied interaction within that entourage, THC and CBD. We touched on how CBD amplified some effects in the Canteen study, but generally, what beneficial role is CBD thought to play when combined with THC?
Yeah, despite that specific finding in the Salience network, CBD is more widely recognized, especially clinically, for its potential to attenuate or reduce some of the unwanted side effects associated with THC. Specifically things like anxiety, paranoia, and maybe some of the memory impairment. By providing this kind of protective effect, CBD essentially acts like a safety buffer. It might allow clinicians or users to administer higher doses of THC to get a therapeutic effect before those negative side effects become too problematic or limiting. So in that sense, it's seen as a key partner that can improve the overall risk-benefit profile of THC.
Right. Now, this brings us to a really interesting term the sources used. The dirty drug reality of plant extracts. That sounds bad, but you're suggesting dirtiness or complexity might actually be a benefit here. How so?
Yeah, dirty drug is an informal term in pharmacology. It refers to a compound that doesn't just hit one single specific target receptor, but interacts with multiple targets throughout the body, a contrast to the highly refined, clean, single-target drugs that pharma often aims for.
Like an atypical antipsychotic, you mentioned olanzapine.
Exactly. Olanzapine is a classic example. It hits dopamine receptors, serotonin receptors, histamine receptors, multiple targets. Now, think about many complex human diseases, chronic pain, multiple sclerosis, maybe certain cancers. These conditions are often multifactorial. They don't have just one single cause or one single pathway that's gone wrong. There are multiple systems involved.
So a single target drug might only hit one part of the problem.
Precisely. And that's why single compound drugs, even though they're the regulatory gold standard, sometimes fail or only provide partial relief for these complex diseases. They can't address all the underlying pathways simultaneously. So the argument is that cannabis extracts, being inherently multi-compound and hitting numerous targets, not just CB1 and CB2, but other receptors like GPR55, TRPV1 channels, may be interacting with enzyme systems. They might actually be better suited for these complex multifactorial diseases.
Because they offer multi-target effects.
Yes. By hitting multiple relevant pathways at once, you could potentially achieve a better overall clinical outcome. And maybe, just maybe, you could do it using lower doses of each individual component compared to trying to max out a single drug therapy, which could minimize the risk of high-dose side effects from any one compound.
That's a powerful argument for embracing complexity. But that same complexity must create huge challenges for figuring out dosing and pharmacokinetics, how the body actually handles these mixtures.
Monumental challenges. It starts right away with just getting the drug into the body absorption. Cannabinoids are very lipophilic. They love fat, hate water. This makes their oral bioavailability notoriously poor. If you swallow an edible or a capsule, studies consistently show THC absorption is really low and variable, maybe only 4% to 20%. For CBD, it's often even lower, around 6%. A huge chunk of the dose gets lost in the gut or destroyed by the liver on that first pass.
Smoking is better for absorption, right? But still variable.
Smoked or vaporized THC does offer higher bioavailability, maybe in the range of 18% to 50%. But even then, there's enormous variability from person to person, even puff to puff, depending on their smoking technique, lung capacity, individual metabolism. It's very hard to get consistent dosing that way.
Okay, now let's hit what might be the single most critical safety issue for any listener who might be taking other medications. Metabolism and the risk of drug-drug interactions. You flag this as high stakes.
This point absolutely cannot be overstated. It's crucial. THC is primarily broken down, metabolized, by a family of enzymes in the liver called the Cytochrome P450 system, or CYP450. Specific enzymes turn THC into its psychoactive metabolite, 11-hydroxy-THC, and then into an inactive form, 11-COOH-THC, which gets excreted. Now, the thing is, this CYP450 system is incredibly important. It's responsible for metabolizing something like half of all pharmaceutical drugs on the market today.
Okay, so where's the danger?
The danger lies with CBD. CBD is a potent inhibitor of several key CYP450 enzymes, especially the CYP3A family, but also others like 2B6, 2D6, and 2C9. Its inhibitory effect on CYP3A is actually similar in strength to powerful pharmaceutical inhibitors like the antifungal drug ketoconazole.
So if someone takes CBD, it slams the brakes on the enzymes that break down lots of other drugs.
Exactly. So imagine you're taking CBD and you're also taking another medication, maybe a blood thinner like warfarin, or an anti-seizure drug, or an immunosuppressant after an organ transplant, and that other drug relies heavily on those specific CYP enzymes, like CYP3A4 or 2C9, to be cleared from your body.
The CBD will slow down the breakdown of that other drug.
Dramatically. If a drug's metabolism is slowed down significantly, its concentration in your bloodstream can shoot up, maybe double or triple or even more.
And that leads to
toxicity. If the other drug has what's called a narrow therapeutic window, meaning the dose that causes toxicity isn't much higher than the dose needed for therapy, this interaction can be incredibly dangerous. It can lead to severe, even life-threatening adverse events. Too much warfarin causes bleeding. Too high levels of immunosuppressants can damage the kidneys.
Right.
This risk is real, and it means anyone taking prescription medications absolutely needs to talk to their doctor or pharmacist before adding CBD or even high CBD cannabis products to the mix. Medical supervision is essential here.
Okay, really critical point. Finally, in this section on complexities, we need to cover a very serious finding related to developmental toxicity, the direct effects observed on human fetal tissue.
Yes, the sources highlighted some concerning findings from studies that directly exposed human fetal testis tissue in a lab setting to CBD, THC, or the combination. The finding was that this exposure was acutely harmful to testosterone secretion. This is obviously a major concern given the documented rise in cannabis use among pregnant individuals in some places.
Was the harm just general toxicity, like killing cells? Or did they identify a more specific mechanism affecting the developing hormones?
It appeared to be quite specific, a molecular disruption of the hormone production process. The effect seemed to involve altering steroid biosynthesis, the way testosterone is made by the specialized Leydig cells in the testes. They used transcriptomics, looking at gene expression, and found a significant downregulation, a turning down of key genes involved in cholesterol transport and steroid synthesis pathways after exposure. Specifically, the gene for a crucial enzyme called CYP17A1, which is essential for making testosterone, was significantly decreased after exposure to the CBD-THC mixture.
Wow.
And on top of that, the combination exposure also seemed to decrease the proliferation, the growth of other important cells in the developing tissue, and increase programmed cell death, apoptosis, within the structures where sperm would eventually develop. So multiple detrimental effects on the viability and function of this developing reproductive tissue.
So this isn't just a vague, it might be harmful warning. It's pointing towards a specific molecular mechanism disrupting essential enzymes for development. That's sobering.
Okay, shifting focus now to the broader societal level. We see this really profound disconnect, don't we, between the long-standing federal classification of cannabis in the U.S. and its rapidly changing legal status state by state.
Yeah, absolutely. The foundation federally is still the Controlled Substances Act from 1970, the CSA, which put cannabis in Schedule, meaning high potential for abuse, no accepted medical use. That's the federal stance. But then you look at the states, and it's a completely different picture. Something like 33 states plus D.C. now have laws allowing medical use. And 11 states plus D.C. allow access for both medical and non-medical or recreational use. It's created this incredibly fragmented patchwork of regulations across the country.
And this rapid legal evolution has outpaced the science in many ways, raising huge unanswered public health questions, particularly around things like workplace safety and driving under the influence. Public health bodies like the CDC have pointed out several really critical gaps in the data we need to actually manage this new landscape effectively.
What are some of the biggest research gaps the CDC is prioritizing? What do we urgently need to know?
Well, first, we just lack good data on the prevalence of cannabis consumption across different industries and occupations. It's hard to assess risk if you don't know who is using what, when, and where. Second, We need much more information on the adverse health consequences, specifically for workers, both acute effects on the job and potential long-term health issues. Third, the link between cannabis consumption and occupational injuries accidents on the job is really poorly quantified right now. Fourth, this is a big one, we urgently need data on the effects on occupational driving. Think truck drivers, bus drivers, people operating heavy machinery. What are the real-world risks? And finally, maybe the toughest nut to crack. We need validated, reliable ways to actually assess performance deficits and impairment from cannabis use in real time, not just detecting the drug, but measuring impairment.
That last point, measuring real-time impairment, that seems to be the absolute crux of the problem for law enforcement, especially with driving under the influence, DUID.
It really is. There's no question that impairment is associated with increasing THC concentration in the body, whether you measure it in saliva or blood. Higher levels generally mean more impairment. However, the unique way inhaled cannabis is processed by the body, its pharmacokinetics, makes enforcement incredibly difficult. When someone smokes or vapes, their blood THC levels peak within minutes very quickly, but then they drop off just as rapidly, often falling by 90% or more within just a couple of hours.
So if someone drives while they're acutely high, feeling the peak effects, but they only get pulled over and tested an hour or two later, their blood level might be way down.
Precisely. It could be significantly lower, potentially even below a legal per se limit if one exists, even though they were functionally very impaired when they were actually driving.
That creates a huge disconnect, doesn't it, between the chemical test result and the actual level of impairment at the time of the offense. Much trickier than alcohol, which clears much more slowly and predictably.
Exactly. Alcohol follows more predictable elimination kinetics. With THC, that rapid drop-off really complicates the legal interpretation of a blood test taken hours after driving. It makes it hard to reliably link a specific THC concentration back to a specific degree of impairment at the time of the incident.
Is there anything in the data about THC levels when other drugs are involved? Does that change things?
Yes, that's another interesting nuance the data suggests. Some studies, for instance, one looking at impaired drivers in Switzerland, found that on average, higher blood THC concentrations were detected when THC was the only drug found in the system. The average was around 8.1 nanograms per milliliter in those cases. But in cases where drivers tested positive for THC plus other substances like alcohol or benzodiazepines, the average THC concentration was lower, around 5.8 nanograms per milliliter.
Why would that be?
Well, one interpretation is that people using only THC might be pushing the dose closer to maximal acute effects, whereas people combining it with other sedating drugs might use less cannabis to achieve a desired effect, or the combined effect leads to impairment at lower THC levels. It just highlights how polydrug use adds another layer of complexity to understanding impairment risk.
Okay, so looking at the research landscape overall then, where is most of the clinical trial work happening right now for potential therapies? And what big picture data are we still missing, especially for long-term safety?
Right now, the vast majority of clinical trials looking at CBD or THC for various neurological conditions, things like chronic pain, epilepsy, multiple sclerosis, they are still concentrated in phase 2.
And phase 2 is about preliminary efficacy, seeing if it works.
Exactly. Phase 2 trials are designed to get that initial signal. Does this compound look like it might be effective for this condition in a moderately sized group of patients? And to gather more safety data, they answer the basic question, does this look promising enough to move forward? The big gap, however, identified in the resources we looked at is the relative lack of phase 4 data. Phase 4 is post-marketing surveillance. That's the research done after a drug is approved and is being used out in the real world by large, diverse populations.
So tracking long-term effects and rare side effects.
Precisely. Phase 4 is absolutely essential for understanding the long-term safety profile, identifying less common side effects that might not show up in smaller trials, and seeing how the drug performs in everyday clinical practice outside the highly controlled environment of a trial.
And without much phase 4 data yet for most cannabinoid products.
Our ability to draw really from conclusions about the long-term safety and effectiveness of widespread cannabinoid use, especially in diverse patient groups or over many years, remains quite limited. That real-world long-term picture is still largely missing. Hashtag tag teleotro.
Okay, so this deep dive has really taken us on a journey, hasn't it? From understanding the endocannabinoid system not just as a drug target, but as this fundamental ancient master regulator of our health, evolved over 500 million years. That's incredible. We saw how acute exposure to external cannabinoids like THC and CBD clearly affects high-level brain function, particularly attention, and how we process what's important. That salience network finding was key. And we confirmed that huge preclinical promise, especially for things like pain relief, but also saw how it's really stalled by this slow clinical translation process and the sheer pharmacologic complexity, that dirty drug reality we talked about in Section 4.
Yeah, we definitely established that natural cannabis is inherently complex, isn't it? It's a multi-compound entity, a dirty drug in that pharmacological sense. It hits many targets and it interacts heavily with our body's own metabolic machinery, like those crucial CYP450 enzymes, creating very real risks for drug interactions that people need to be aware of. And all of this, it really leads us to a final provocative thought, something to maybe chew on that ties this whole deep dive together. Think about this. A central ongoing challenge in developing new medicines, especially for complex chronic diseases like pain or cancer or neurological disorders, is finding those single, clean molecular targets that actually work effectively. It's really hard. Pharma spends billions trying. So the question becomes, given that difficulty, are we perhaps limiting ourselves by so strictly insisting on the pure single molecule approach that the current regulatory environment strongly favors?
Especially when, as you pointed out, the body's own ECS seems built for multi-target regulation, using molecules like 2-AG that hit multiple receptors and are designed for quick, localized action.
Exactly. So could it be that the controlled complexity we see in a whole plant extract, or maybe in carefully engineered designer combinations that embrace polypharmacy instead of shunning it, could that actually be the key to unlocking the future of cannabinoid therapeutics?
It forces us to think differently, doesn't it? To move beyond just the traditional find one target, hit it with one pure drug paradigm, and instead consider whether a kind of organized, controlled complexity might be more effective for complex problems.
It's a really compelling thought, isn't it? That the very pharmacological complexity of the plant, which makes it messy for traditional drug development, might actually reflect the kind of multi-system strategy the body itself uses and perhaps needs for complex regulation and healing. It potentially turns what sounds like a marketing slogan, the entourage effect, into a concept with deep, sophisticated scientific implications.
A lot to think about there. Thank you for joining us for the deep dive. We hope you leave here feeling well-informed and maybe pondering the future implications of embracing multi-target therapy versus the single-target focus in the ever-evolving world of medicine.